Hi everyone, and welcome to Biology Professor. Today we're going to go over how to conduct a most probable number test. So a most probable number test, or MPN, is sometimes, that's how the method is referred to as the MPN method. This is a method that is used to count bacteria, to measure the amount of bacteria in a sample. And it is ideal for estimating the number of microbes in very dilute samples. So when we say very dilute samples, we mean samples where the bacteria are not very concentrated. So we're not talking about lab plates or lab cultures where you can do a plate count or an optical density. These are samples where there are much fewer bacteria present, or at least that's what you suspect. An example of this would be various kinds of water and food testing. So for example, testing certain kinds of water sources for coliform bacteria. Coliform bacteria are bacteria that can ferment lactose, and we have a lot of coliform bacteria in human feces. And so if there's coliforms in water, that's typically indicative of fecal contamination. So the way the MPN method works is it uses changes in either color of broth or changes in turbidity, that's the cloudiness that you see when bacteria are growing in liquid culture. So it looks for either color change or turbidity change to detect bacterial growth. And then you look at how many tubes either changed color or have noted turbidity, and you can count those tubes and then interpret your results by comparing your results to a standard reference table. And we'll go over an example of that in a second. But when you are using the standard reference table, it's important that you realize that the reference table will give you a number, a most probable number, an MPN, and that an MPN is equal to 1 CFU. CFU is a common way of referring to bacteria, and it stands for colony forming unit. And basically that just means one viable cell. So just to recap, this method gives you a number, a most probable number for how many bacteria are in a certain sample, and that equates to one colony forming unit, which is the same thing as saying one viable cell. So one living cell, one cell that's capable of reproducing. So let's go through an example. First, you have your inoculation. And so in this example, we're looking at a water sample. Perhaps it's from a pond, perhaps it's from like a recreational swimming area, perhaps it's a drinking water sample. We have some water sample here. And then we have 3 sets of 5 lactose broth tubes. And remember that in this example, we're looking for coliform bacteria, and coliform bacteria can ferment lactose. You could do this method with other kinds of tests where you're looking for a different kind of color change or a different growth and turbidity in different kinds of broth, depending on the type of bacteria you're looking at. So here we have our 3 sets of 5 lactose broth tubes. And what we do is we take 10 milliliters from the drinking water sample and inoculate each of these 5 tubes. So 10 milliliters added to each one. Then we take 1 milliliter of the water sample and add it to each of these tubes. And then we take 0.1 milliliters of water from the sample and add it to each of these tubes. And so we're having this 10-fold decrease each time in the amount of water that's being added, and so theoretically in the amount of viable coliforms that are being added to these tubes. Then we incubate overnight at 37 degrees. Typically you want to incubate for like 24 hours for this particular method. Then it's time to read results. And what that means is you're looking for the color change. So when coliform bacteria are present in these broth tubes, they will ferment the lactose, and that results in a color change. And so for this particular test, we have 4 positive tubes here. So 4 of them showed a color change, 1 did not. We have 3 positive tubes here. 3 showed a color change, 2 did not. And then where we were adding the smallest amount of the water sample, we've just got 1 positive tube. So 1 changed color and 4 did not. And so that is our results that we then want to come and compare in the reference table. So our pattern, if you will, was 4, 3, 1. And so we look at our reference table. If you're a student, this is often in the back of a textbook or in the back of like a lab manual. So you look for the pattern of 4, 3, 1. And this tells you that if you see the 4, 3, 1 pattern, the MPN, the most probable number of coliforms in the original sample, was 33 per 100 mils. Now, if we had gotten different patterns up here, if it had been 4, 4, 1, or if it had been 5, 2, 1, different patterns will result in a different most probable number. But with this pattern of 4, 3, and 1, the best answer statistically is that for every 100 milliliters of that sample, we have 33 coliform bacteria. The table also gives us a 95% confidence range. And for the pattern 4, 3, 1, the 95% confidence range is 11 to 93. So the most probable number is 11 to 93 per 100 milliliters. And all this means is that whereas 33 is like the best guess, the most sort of statistically supported guess, you can be 95% confident that in this water sample that gave this pattern, there is somewhere between 11 and 93 viable coliform bacteria per 100 mils of this sample. So that is how you conduct an MPN test. If you want to learn more about bacterial growth, you can check out my video on bacterial growth curves and binary fission. But that's it for today. Thanks for watching, Biology Professor.